picture a plant for a second. You have almost certainly seen it growing wild, like on the side of a highway or just in some empty dirt lot.
Yeah, the ones people are always trying to pull out of their gardens.
Right, exactly. It's tall, sometimes pushing like 10 feet, and it has these fiercely prickly leaves, thick, spiky stems, and these striking, bright, purple flowering heads.
Very distinct looking.
And if you look closely at the leaves, you see these milky white veins running all through them. It's milk thistle, basically a common weed.
Very persistent weed.
Yeah, and people have just been walking past it, ignoring it, or, occasionally throwing it into traditional remedies or salads for well over 2,000 years. But here is the question we're exploring today, and it's a big one. What if this ancient thorny plant holds a vital blueprint for modern oncology?
I mean, it sounds almost like science fiction, but the clinical data we're looking at is very, very real.
Welcome to today's deep dive. If you are sitting there wondering how a roadside weed connects to cutting-edge cancer research, well, our mission today is to unpack a comprehensive 2022 scientific review.
Right, from the Iranian Journal of Basic Medical Sciences.
Exactly. And this review focuses entirely on milk thistle, scientifically known as Silymarin, and specifically its active chemical constituent, which is called Silymarin.
Silymarin, yeah.
We are going to explore how this ancient folk remedy is currently driving this fascinating wave of modern patents for cancer prevention and treatment.
It's really bridging the gap between historical botany and rigorous clinical science, which for anyone listening, whether you're a health enthusiast or just curious about how ancient remedies translate into modern medicine, this is basically a masterclass in how basic science can validate traditional knowledge.
So before we jump into the cancer patents, we probably need to understand what this plant actually is, its historical resume.
Well, to really grasp how we got to modern cancer patents, we have to start with that history. Milk thistle has a documented 2,000-year history in traditional medicine.
2,000 years?
Yeah. You find it in foundational historical texts like the ancient Greek pharmacopoeia, al-Hashish Dioscorides.
Oh, wow.
Yeah, and it was meticulously documented in there under the name Harshbari.
I always find it fascinating how our ancestors mapped out the uses of these plants purely through, centuries of trial and error. What were they primarily using it for back then?
Mostly the liver.
Yeah.
Historically, it was heavily utilized for liver disorders. So jaundice, hepatitis, early cirrhosis.
Okay, so liver stuff mostly.
Mostly. But it went beyond standard liver disease. It was actually the go-to treatment for severe poisoning.
Wait, like what kind of poisoning?
Like snake venoms.
Oh, wow, really?
Yeah, and mycotoxins, which are these highly lethal toxins produced by fungi. They even use the seeds as a galactagogue, which means giving it to breastfeeding mothers to help stimulate milk production.
Okay, that is quite the resume. But, modern biomedical science isn't just handing out patents because an ancient Greek text says a plant is good for you.
Right. No, to get a patent, you have to isolate the active biochemical engine.
Which they did, right.
They did exactly that.
Yeah.
So the primary active component in milk thistle, which is concentrated mainly in the leaves, the seeds, and the fruits, is this complex called silymarin.
Silymarin, right.
And when you break silymarin down, it's made up of about 70 to 80% of a specific class of compounds called flavonolignin.
Okay, so when I hear a word like flavonolignin, I try to visualize what they are actually doing in the body. If they're protecting the liver from something as aggressive as snake venom or fungal toxins, are they acting like a microscopic sponge, just sort of soaking the poison up?
It's actually more structural than a sponge.
Yeah.
Think of flavonolignin as membrane stabilizers.
Membrane stabilizer.
Right. They physically alter the outer lipid layer of the liver cells so that toxins simply cannot bind to the cell wall and penetrate the interior.
So instead of hunting the toxins down, the flavonolignin are basically changing the locks on the cell doors.
That's a great way to put it. The toxin is floating around in the bloodstream, but it can't get inside the liver cell to cause damage.
That makes total sense.
And within that group of, lock-changing flavonolignin, there is one absolute superstar molecule called silibinin.
Okay.
When you look at the major therapeutic effects of milk thistle in these clinical trials, silibinin is the one doing the heavy lifting.
All right, I have to push back here for a second with a healthy dose of skepticism because, the dietary supplement industry is notoriously underregulated.
Very true.
We hear about miracle herbs constantly. So if I go to the store, buy a highly concentrated milk thistle extract, and just take a massive dose every day, am I actually doing more harm than good? What does the hard clinical data say about toxicity? Because, I mean, even water is toxic if you drink enough of it.
Right. But the clinical data on safety is actually one of the main reasons oncologists are so interested in it. Silymarin is incredibly safe for human consumption.
Really? Even at high doses?
Yes. In clinical trials, researchers have tested very high doses. We're talking 700 milligrams taken 3 times a day for a full 24 weeks.
Wait, so over 2,000 milligrams a day for 6 months?
Exactly. And even at that sustained high level, researchers found no significant liver, kidney, or systemic toxicity.
That's wild. No side effects at all.
Well, the only negative effects reported across the board were very minor gastrointestinal discomforts.
Like an upset stomach.
Yeah, mild nausea or upset stomach, and only in a small subset of patients.
Okay, but what about drug interactions? Because most pharmaceutical drugs are processed in the liver by this enzyme system called cytochrome P450, right?
Right, the CYP450 pathway.
So if Silymarin is heavily interacting with the liver, doesn't it jam up those enzymes and prevent other medications from working?
You'd think so, but it surprisingly has a very low drug interaction profile. It leaves the cytochrome P450 system mostly alone.
Oh, interesting.
Yeah, it doesn't severely induce or inhibit those enzymes, which means it doesn't hog the liver's processing bandwidth.
Which means it plays nicely with other medications.
Precisely.
Okay, so that naturally brings us to the liver cancer patents. Hepatocellular carcinoma, or HCC. That's the primary type of liver cancer.
And it is a massive global threat. It currently stands as the 3rd leading cause of cancer death worldwide.
Wow. 3rd.
Yeah, and the treatment landscape is incredibly difficult. I mean, even for patients who are diagnosed early enough to undergo surgery, the 5-year survival rate is deeply concerning. It hovers around like 47 to 53%.
Because the recurrence rate is just terribly high.
Exactly.
So you have this incredibly aggressive cancer in an organ that is already responsible for filtering the body's blood. How does Silymarin intervene in that kind of environment?
Well, it operates with a dual action. It plays defense for the healthy cells and offense against the cancerous cells.
Okay, break that down for me. Start with defense.
On the defensive side, the review highlights a study on breast cancer patients who are undergoing a really hard chemotherapy regimen known as ACT.
Let me jump in on ACT for a second because that is a combination of doxorubicin, cyclophosphamide, and paclitaxel. In the oncology world, that is basically heavy artillery.
It really is.
It's a carpet bombing approach to kill the breast cancer, but those specific drugs are notoriously brutal on the liver. They cause severe hepatotoxicity.
They do. And that toxicity often forces doctors to lower the chemotherapy dose, right?
Yeah.
Which gives the cancer a chance to survive.
Right. You have to back off the drugs so you don't destroy the liver.
But when these patients were given Silymarin alongside their ACT chemo, it significantly reduced the severity of that liver damage.
Oh, wow.
Yeah, the Silymarin basically threw a protective shield around the healthy liver cells, allowing the patients to tolerate the full dose of the chemotherapy.
That's the defensive side. What about the offense, like against the actual liver tumors?
In animal models with induced liver cancer, Silymarin physically reduced the total number and multiplicity of macroscopic hepatic nodules.
The visible tumors.
Right, visible tumors in the liver. It also powerfully downregulates COX-2, which is an enzyme that acts as a major marker of inflammation.
And a driver of tumor growth.
Exactly.
Yeah.
But the most interesting mechanism happens at the genetic level. It involves these proteins named P53, BAX, BCL2, and survivin.
Okay, let's translate that protein soup into a physical mechanism. So in our healthy cells, we have this biological process called apoptosis, right, which is programmed cell death.
Right.
Essentially, every cell has a built-in self-destruct button for when its DNA gets corrupted or damaged beyond repair. But cancer cells survive by cutting the wires to that self-destruct button.
Exactly. They overexpress anti-apoptotic proteins like the BCL2 and survivin we mentioned to basically jam the signal so they can keep multiplying forever.
So they just ignore the destruct command.
That is the core pathology of a tumor. So what Silymarin does is upregulate the pro-apoptotic proteins, the P53 and BAX, while simultaneously downregulating the anti-apoptotic ones.
So Silymarin is acting like a molecular electrician. It comes into the cancer cell, bypasses the tumor's jamming signal, and physically rewires the self-destruct button so the cancer cell can finally recognize its own damage and destroy itself.
The biochemistry supports that analogy perfectly, yeah.
Oh.
And to ensure the cancer cell doesn't multiply while the rewiring is happening, Silymarin forces what we call cell cycle arrest at the G0-G1 phase.
Meaning it pulls the emergency brake before the cell can even copy its own DNA to divide.
Exactly. It just freezes the cell. And the crucial takeaway here is that Silymarin does all of this anti-proliferative work without harming the non-tumor healthy liver cells.
That's incredible.
It is. Inventors are using this exact mechanism in patents formulated to promote liver regeneration and reduce viral loads in hepatitis. They're trying to stop chronic damage early and prevent the environment that allows hepatocellular carcinoma to develop in the first place.
Well, treating the liver makes intuitive sense because the liver is designed to process whatever we ingest. But the review pushes out into organs that don't have that built-in filtering capability, right?
Right.
Starting with the pancreas. And pancreatic cancer is just notorious for hiding until it's very advanced.
It's extremely malignant, yeah, with a very poor prognosis. But the research on silibinin, that superstar molecule we talk about, shows it inducing DNA damage specifically inside the pancreatic cancer cells.
Inside the cancer cells themselves.
Yes. And it also dramatically reduces the expression of a gene called CMYC.
Let's pause on CMYC for a second because throwing around gene names can get confusing for you listening at home. What role is CMYC playing for the pancreatic tumor?
Think of CMYC as the master throttle for cancer metabolism.
The throttle, okay.
Right, because cancer cells need massive amounts of energy, mostly glucose, to fuel their rapid, uncontrolled growth. CMYC is the signal demanding that energy.
So by reducing its expression, silibinin's effectively starving the pancreatic cancer cells. It's putting a restrictor plate on the tumor's engine.
Which brings us to the prostate cancer patents. Prostate cancer is the most prevalent non-skin cancer in men globally. We're talking over 1.6 million new cases diagnosed every year.
It's a huge issue. And the review highlights this fascinating clinical trial for prostate cancer, where men were given 570 milligrams of Silymarin daily for 6 months.
And what happened?
Well, by the end of the trial, they saw a significant reduction in PSA levels, or prostate-specific antigen, which is the primary blood biomarker used to track how aggressive the cancer is.
Oh, wow.
Yeah, and they also recorded lowered levels of LDL and total cholesterol, which are serum lipid markers that are heavily associated with the progression of the disease.
Okay, but how is it slowing the prostate cancer down? Like, what's the actual mechanism there?
One of the primary ways is by inhibiting the secretion of proangiogenic factors. Angiogenesis, which is the creation of new blood vessels. Because a tumor can't grow past a few millimeters in size unless it tricks the body into building fresh blood vessels, right, to supply it with oxygen and nutrients.
Right, the tumor needs a blood supply.
So the tumor is trying to lay down plumbing, and Silymarin is basically cutting the pipes.
Exactly. The Silymarin disrupts capillary tube formation, so the new vessels just fail to form. But it actually goes a step further by inhibiting specific chemokine receptors, most notably one called CXCR4.
Is CXCR4 another growth signal?
It functions more like a homing beacon for metastasis.
Oh, I see.
Yeah, when prostate cancer cells express high levels of CXCR4, they're primed to break away from the main tumor, enter the bloodstream, and invade other tissues, particularly bone tissue, which is where prostate cancer often spreads.
Right. So by blocking that specific receptor, silibinin helps lock the cancer in place. It prevents it from migrating.
That's the idea, yes.
Okay, so I understand the mechanism, but here's my pushback on the actual patents themselves. I'm noticing this really distinct pattern in the patents for the pancreas, the liver, and the prostate. The inventors are almost never giving patients pure, isolated milk thistle extract. Aren't. For the pancreas, they combine it with melatonin, zinc, and quercetin. For the prostate, the review mentions patents mixing Silymarin with turmeric, broccoli extract, alpha-lipoic acid, and aloe vera.
Yeah, it's quite the mix.
Right. So if Silymarin is this capable on its own, you know, starving the engine by dropping CMYC, cutting blood supply, blocking metastasis beacons, why are the patents mixing it into this massive botanical soup? It feels counterintuitive. If you have a star player, why dilute the dose with, like, broccoli extract?
It's a fair question, but you are touching on a foundational challenge in oncology here, which is cancer's sheer adaptability.
Adaptability.
Yeah. Cancer cells are notoriously plastic. If you block just one pathway, say you successfully inhibit that CXCR4 receptor with Silymarin, the tumor will often mutate and upregulate a completely different chemokine receptor to escape and metastasize anyway.
So cancer is like a river. If you dam one pathway, it just builds pressure and overflows into another route?
The data backs that up completely. That is the underlying theory of synergy in phytomedicine.
Synergy.
Right. By combining Silymarin with other non-chemotherapeutic agents, you create a multi-pronged siege strategy.
Oh, I get it. So the curcumin found in turmeric might suppress a specific inflammatory pathway that Silymarin misses entirely.
Exactly. And the sulforaphane in the broccoli extract might trigger a completely different cellular antioxidant response. You're hitting the cancer's metabolism, its blood supply, and its inflammatory pathways simultaneously.
Blocking multiple escape routes so the tumor cannot easily mutate around the treatment.
Precisely.
But doesn't mixing all these active compounds increase the risk of unpredictable interactions in the body?
It can, which is exactly why these specific combinations are patented based on rigorous clinical testing. Furthermore, these combinations solve a major pharmacokinetic problem, which is bioavailability.
Right, because Silymarin is a large molecule.
Yes, and it's not always absorbed perfectly through the human gut lining. So coupling it with certain lipids or other specific plant compounds acts as a carrier system. It ensures the Silymarin actually reaches the target tissue in the prostate or the pancreas.
That makes perfect sense. Building a botanical cocktail where every ingredient covers the biological blind spots of the others.
Exactly.
Okay, so we have spent our time deep inside the body, the liver, pancreas, and prostate. But the review highlights a completely different application regarding our body's outer shield, the skin.
Yes.
And here, the research shifts gears away from oral capsules and focuses entirely on topical creams.
The topical application data is actually some of the most compelling in the review. When we look at skin cancer, the primary external driver is solar ultraviolet radiation, specifically the UVB rays.
And UVB rays physically damage the DNA in our skin cells.
They do. They cause specific structural lesions in the DNA called cyclobutane pyrimidine dimers, or CPDs.
Okay, so the word dimer means two molecules linking together, right? So the UV light hits the skin and literally melts adjacent localized pieces of our DNA together that shouldn't be attached. It creates a physical kink in the genetic code.
A structural kink is a great way to visualize it.
Right.
And if the skin cell tries to replicate its DNA with that kink still there, it introduces a permanent mutation, which is the genesis of melanoma or carcinoma.
Wow.
And what the clinical research shows is that silibinin accelerates the repair of these dimers. It fundamentally changes how the cell handles the radiation damage.
So it's not acting like a traditional sunscreen then? Like it isn't just sitting on the surface of the skin bouncing the UV light away?
No, not at all.
It's functioning like a molecular mechanic, rushing in to unkink the DNA strands after the UV damage has already occurred, basically fixing the engine while the car is still running.
That internal repair mechanism is incredibly potent. And where this topical application truly proves its value in oncology is in the treatment of radiodermatitis.
I read the clinical study in the review on this, and honestly, the numbers are staggering.
They really are.
For you listening, radiodermatitis is essentially severe radiation burns. Patients undergoing radiotherapy for tumors often suffer these burns, which are agonizing and severely degrade their quality of life. But how do you even measure the success of a topical cream in that scenario? Like, what's the clinical metric?
Oncologists grade the skin toxicity on a scale. Grade 1 is mild redness. Grade 2 is patchy, moist peeling of the skin, and moderate pain.
Okay.
Grade 3 is severe, widespread peeling, ulceration, and intense pain. In one clinical study, breast cancer patients undergoing a 5-week course of radiotherapy were monitored. Half were given a standard care protocol, and the other half were given a Silymarin-based cream to apply to their chest.
And after 5 weeks of targeted radiation, what happened?
In the control group, so the patients not using the Silymarin cream, 52% of them developed grade 2 skin toxicity.
Over half the patients suffered moderate, painful peeling.
Right, but in the group using the Silymarin-based cream, only 9.8% developed grade 2 toxicity.
That is a massive reduction. What about the severe grade 3 burns?
It was 28% in the control group, compared to just 2% in the Silymarin group.
2% versus 28%. That's the difference between a patient being able to wear clothes and sleep somewhat comfortably versus being in agonizing constant pain.
Exactly. It's a game changer for quality of life.
And these topical patents go beyond just radiation treatment, too. The review mentions a patent out of South Korea dealing with air pollution.
Yeah, there's a cosmetic and therapeutic patent that uses Silymarin to protect the skin against severe urban air pollutants. Specifically, a highly toxic compound called dioxin.
Dioxin, okay.
The Silymarin formulation penetrates the skin and literally prevents the expression of genes like CYP1A1 and COX-2.
And those are the specific genes triggered by the dioxin to cause cellular inflammation and rapid skin aging, right? So the cream is preemptively shutting down the skin's inflammatory overreaction to the smog, which, I mean, it really highlights Silymarin's value as a complementary therapy because cancer treatments like chemotherapy and targeted radiation are brutally toxic by design. They have to be to kill the tumor.
They do.
But by applying something like Silymarin, it allows the patient to actually endure those aggressive treatments by drastically improving their physical tolerance.
And that physical tolerance is crucial. It extends to the most pervasive side effect of all. Which brings us to our final point from the review.
Yeah.
Cancer-related fatigue.
This is such an important point because fighting cancer isn't just about attacking rogue cells under a microscope.
No, it's not.
It's about the lived experience of the human being who is carrying the disease. The treatments, the persistent fear, the physiological toll, it all culminates in this crushing systemic exhaustion. And this isn't just normal tiredness where you didn't sleep well. It's a bone-deep exhaustion that sleep simply does not fix.
Right. The clinical term is cancer-related fatigue, and it affects anywhere from 25% to 99% of cancer patients. For a long time, it was dismissed as purely psychological, just a byproduct of depression or stress.
But it's not.
No. Researchers now know it is a biological mechanism driven by inflammatory cytokines.
Okay, so cytokines, these are the same signaling molecules the immune system uses to fight off, like a severe flu, right? The ones that make your muscles ache and force you to stay in bed.
Exactly. But in a cancer patient, those cytokines are constantly flooding the brain, creating this permanent neurochemical fog.
The central nervous system is essentially under constant inflammatory assault.
Right. And to combat this, the review highlights a specific patent developed in Russia. The inventors created a composition using extracts of common ginseng, pharmaceutical ginger, and milk thistle.
Okay, so another combination.
Yes. And they evaluated this botanical cocktail on male patients with primary tumors in the prostate, pancreas, lung, and colon.
And just to clarify, these were patients experiencing chronic fatigue who had very limited life expectancies, correct? Like they were dealing with very poor prognosis.
They were. And when administered this composition, the patients showed a notable, measurable improvement in overall vitality. Their daily mood improved, their appetite returned, and they even reported a decrease in peripheral pain.
Wow. And that's because the Silymarin, working in tandem with the ginger and ginseng, is lowering that systemic inflammation and clearing the cytokine fog out of the central nervous system.
Exactly.
It treats the lived experience of the patient. It gives them their quality of life back in the time they have.
And really, knowledge in medicine is only as valuable as its practical application.
Yeah.
Lowering systemic inflammation doesn't just make the tumor smaller. It fundamentally changes the patient's daily reality.
Absolutely. Well, let's bring this all home. Today, we have unpacked the hidden potential of a 2,000-year-old spiky weed. We've explored how milk thistle, driven by its active engine Silymarin, is currently generating modern medical patents across the globe.
It's pretty amazing.
It's a chemopreventative agent that acts as a molecular electrician, rewiring corrupted cancer cells to self-destruct through apoptosis. It cuts off the blood supply to tumors, acts as a molecular mechanic to unkink UV-damaged DNA, and makes the brutal realities of modern chemotherapy and radiation deeply more bearable for the patient.
But for you listening, the ultimate takeaway is not that Silymarin is a standalone magical cure for cancer. It is not.
Right.
Rather, it's a brilliantly validated example of how nature provides us with sophisticated, multi-targeted molecules. These are tools that can and should be integrated alongside conventional modern medicine to treat the entirety of the disease while simultaneously treating the entirety of the patient.
It really bridges the gap between the ancient apothecary and the modern oncology laboratory, which leaves you with a final thought to mull over today. If a thorny weed with milky veins, one we have been walking past, ignoring, and actively weeding out of our gardens for 2 millennia, holds the exact biological blueprint for repairing radiation-damaged DNA and mitigating the harshest side effects of modern chemotherapy. What other invisible, annoying weeds are currently growing in our backyards, just waiting to unlock the next major medical breakthrough?
It's a fascinating question to end on.
It really is. Thanks for joining us on this deep dive. We'll catch you next time.